Hello everyone, I am Sumit Walia, a fourth year PhD student at UCSD, working with Professor Yatish Turakia, and today I will present our work on building ultra-large pangenomes using scalable and comprehensive techniques. I'll start by introducing pangenomics, its applications, and the limitations of current representations. Then I will introduce PanMANs, a novel pangenome format that we developed, which improved the representation power of pangenomic formats, yet it's most compact and scalable. Then I'll describe its various contributions and interesting results, and end my talk with conclusion and future work. Let's get started. So pangenomics is an emerging field that deals with a collection of genomes from a single species, and this collection of sequences that belong to the same species is called a pangenome. Pangenomes enables researchers to view the complete genetic diversity within a population, and with the availability of this view, pangenomes help overcome the issue of reference bias that arises with a single linear reference sequence, as a single genome does not and cannot represent the whole genetic diversity. And this field is fueled by the advent of advancements in technologies, particularly low cost and high throughput sequencing technology that has enabled researchers to sequence genomes from the same species at an unprecedented rate. For example, during COVID-19 pandemic, more than 16 million SARS-CoV-2 genomes were sequenced, and the trend suggests that these numbers will further grow in the future. Pangenomics has not only overcome the issue of reference bias, but also has enabled applications in various fields. The genetic variations has enabled researchers to study diseases in humans. For instance, the human pangenome has enabled the detection of structural changes that are found to be associated with diseases such as autism and schizophrenia. Not only that, these genetic variations have also enabled study desirable traits in plants and animals. For example, in one of the pangenome tomato studies, researchers uncovered variations associated with tomato flavor traits and pinpointed them to improve fruit flavors. Similar studies were performed with the cow's pangenome to study the correlation of variations with milk lactose content. In pathogens such as Staphylococcus aureus pangenome study, researchers found genes associated with antimicrobial resistance, and these regions affected by structural variations, helping explain why certain variations have succeeded globally. These variations also improve metagenomic studies. For instance, WEP, which is a wastewater-based epidemiology tool that uses variations present in samples collected from wastewater for surveillance, such as detecting circulating and novel variants. Pangenomes serve as an important tool for these studies too. And finally, it improves the read mapping and transcriptomic efficacy too, due to the presence of complete set of genetic variations during evaluations. Now that we have learned about pangenomics and its applications, we will delve into various pangenomic representations. It is important to realize that the data structures that are used for pangenomic research are critical because they not only determine the efficiency of representation and supporting applications, but also the representative power itself. The most widely used pangenomic representation is graph-based. Essentially, given a set of sequences, the region without variation is represented as a single node in the graph, and variations create alternative paths or bubbles in the graph. Each path in the graph corresponds to a sequence in the collection. This graphical representation has many alterations. GFA, which is a human readable format, VG, a serialized one, with the option to add small indels, and GBZ is developed to improve the memory efficiency. With these advancements, graph-based representation have achieved excellent reduction in reference bias, has enabled various applications using improved read mapping. However, there are few limitations of these representations. These representations do not store evolutionary and mutational histories. They only store variations which is shown on the top right figure. And these formats are inefficient in terms of storage. To illustrate this, I have added a table at the bottom right that shows the file sizes of different formats for various pathogens. The file sizes can be significantly improved as we will see in later slides. Then there are some formats that have been recently developed which focus on data compression such as AGC and Minify. And then there are other set of formats that try to improve the representation power of the existing formats, such as pangraph, try to extend the representation power of graph pangenomes by incorporating tree topology. Similarly, TS-Kits adds improvement by representing recombination events using tree sequences. With all these formats, we essentially have two sets. One that focuses on compression and the other that focuses on representation power. And this work with PanMANs, we are able to achieve both high compression and better representation power than existing formats. Now, before discussing PanMANs, I would like to introduce the key idea behind this work, which is called evolutionary compression. It was first demonstrated through a data structure called MAT, which stands for Mutation-Annotated Tree. MAT stores inferred phylogeny or tree structure, a root sequence, and the mutations inferred on each branch as shown in the figure. The nice property about this format is that any sample at the tip or the internal node can be derived from the root sequence by applying mutation on its path from the root. This format was first used in Usher, and Usher MAT had a few limitations. First, it has reference bias because the mutations are inferred with respect to a reference sequence. Secondly, it only stores substitutions and indels were not represented. And many works has demonstrated that indels could be critical for many studies. For instance, indels are proven to be crucial member of lineage defining mutations in SARS-CoV-2 phylogenies. Finally, it cannot represent recombination and horizontal gene transfer events, or the events that break the vertical mode of evolution and form network-like connections as shown on right, as it assumes that genetic material is transferred from a single parent. These are the limitations of AssureMat, and to overcome these limitations, we have developed PanMAN, which stands for Pangenome Mutation-Annotated Network. That is a novel pangenomic representation as well as a data structure which uses evolutionary compression to represent a collection of genomes. It is the first format that unifies alignment, phylogeny, into a single format. The preprint can be found on bioRxiv and it currently is under review in Nature Genetics. Now we'll see how PanMAN overcomes the limitations of previous works. First, indels are added to MAT, and we call this format as PanMAT, which stands for Pangenome Mutation Annotated Tree. We achieve this by using multiple sequence alignment as input and considering gaps as special character. An ancestral reconstruction algorithm is used to infer the root sequence and mutations. Hence, multiple sequence alignment defines the coordinate system and overcomes the issue of reference bias that MAT has. Finally, PanMAT stores the phylogeny, mutations, and the root sequence. Next, PanMAT also represents structural changes and rearrangements. To do that, first identified homologous blocks are used as input. These blocks are aligned, and again an ancestral reconstruction algorithm is used to infer the mutations. Here, a pseudo root is stored, which is a linear ordering of all the blocks in the PanMAT. Hence, a PanMAT stores the tree structure, block mutations, and pseudo root. And for each block, nucleotide mutations are stored that were shown on the previous slide. Finally, PanMAT is extended to PanMAN, which is Pangenome Mutation-Annotated Network, to represent complex mutations such as recombinations and horizontal gene transfer. These mutations break the vertical mode of evolution and form a network-like structure. To handle this, PanMAN connects multiple PanMATs together through network edges, and these edges, which are shown in red dotted lines here, store the complex mutation information such as breakpoints and node IDs. And this is how PanMAN represents complex mutations. In summary, PanMAN extends the representation power of pangenomes from just storing variations to storing mutations and evolutionary information. And being the most information-rich format, it is still the most compact and scalable. That we'll see now. Now I'll describe the important contributions and results of this work. As we saw, PanMAN extends the representation power of pangenomes and remains the most compact format across various pathogens that we evaluated. It includes viruses and bacterial datasets. Overall, PanMAN provides up to 680 times compression over GFA, 221 times over BG, 42 times over GVZ, and 14 times over PanGraph. Also, we observe that the scalability of PanMANs remains better than any other format across all pathogens. And to further support this argument, we are very excited to share that we have built the largest pangenome with SARS-CoV-2. This PanMAN contains more than 8 million genome sequences and the file size is only 366 MB. It provides more than 3000 times compression over its multiple sequence alignment. To build this PanMAN, we used Usher-Mat phylogeny, the tree structure, and used Twilight to construct the multiple sequence alignment. Twilight is a new multiple sequence alignment tool capable of constructing MSAs for very tall and wide alignments. Tall meaning having large number of sequences and wide meaning long sequences. Twilight is accepted and presented in ISMB 2025 and published in Bioinformatics. Please scan this QR code to learn more about this tool. Further, we analyze the quality of PanMAN by comparing its substitution landscape with Usher-Mat, and we observed high concordance between both of them as shown on the plot on top right. Next, we analyze the indel landscape of the PanMAN. Again, this is the first time that an indel landscape is evaluated for SARS-CoV-2 at this scale. We analyze indels in the spike region. Since the S2 subregion in SARS mediates membrane fusion between the virus and the host cell, therefore prone to fewer mutations in the region. We found similar landscape in PanMAN as shown in the bottom right figure, that is, dominant indels in the S1 subunit and almost no indels in the S2 subunit, which matches with the previous literature. These results provide strong evidence of high quality of the PanMAN. Also, we represented 367 recombination event in the PanMAN, which was inferred using Rivet. This is just a snapshot of the complete network. This large PanMAN and other PanMANs generated in the study is publicly available online at this link. Last but not least, we built PanMAN utils. Although PanMAN has its benefits with crucial information embedded in it, it is important that users can extract that information and use it for downstream applications. To smoothen that process, we created a software with a suite of functionality and call that software PanMAN utils. It can be used to build PanMANs, modify them, and extract information from them. PanMAN utils takes alignment and a phylogeny as an input to build a PanMAN and supports pangraph, multiple sequence alignment, and GFA as an input. Thanks to the pangraph tool, as most of the PanMANs are built using pangraph, except the 8 million one, as it helps to construct the most compressed PanMANs for the datasets we evaluated. A few functionalities of PanMAN utils include raw and multiple sequence alignment extraction in FASTA format. Users can also extract only a snippet of MSA, a region of evaluation if the complete MSA is not needed. PanMAN utils also allow users to extract multiple whole genome alignment in map format, extracting the alignment of homologous blocks. To support interoperability, PanMAN utils can convert a PanMAN into a GFA and vice versa for smooth integration with existing tools and pipelines. Another interesting feature of PanMAN utils is that it can extract variations from a PanMAN with respect to any sequence as reference. For example, here the variations are with respect to S2. It is possible because PanMAN stores multiple sequence alignment, hence computing variations with respect to any sequence becomes trivial. Finally, it also supports many other functionalities such as summary extract, amino acid translations, etc. In conclusion, we present PanMAN, a novel data representation that improves the representation power of pangenomes. It is the first format that encodes phylogeny, mutations, and alignment in a single format, and yet remains most compressed format. It provides excellent compression up to 680 times compared to existing formats. Again, it is highly scalable and enables the construction of largest pangenome containing 8 million SARS-CoV-2 genomes. We also present PanMAN utils to build, modify, and extract information from PanMANs. In future, will represent more complex genomes such as of eukaryotes in PanMANs and accelerate the construction step using advanced modern GPUs. Finally, I would like to thank my advisor, Professor Yatish Turakia, other co-authors, collaborators, and funding agencies for the support. Thank you for listening.